This is the AQA AS Biology Paper 2 from 2018. Some general comments on this paper that were on the examiner's report included that students' knowledge was very limited when it came to digestion, prokaryotic DNA and evolutionary relationships. And they were weak on exam skills when giving descriptions on evaluating or comparing and contrasting. And also the way that students made use of the information in graphs and tables weren't particularly well shown. And quite a few students didn't show an understanding when it came to diluting solutions. But I'll go over these points in detail when we get to relevant questions. Question 1 asks us to identify some cell structures from the descriptions given. So the first one we've got is stacked membranes arranged in parallel and contains DNA. So the key terms in this one are they have stacked membranes and they contain DNA. So the stacked membranes should point you towards the chloroplasts in this case. The answer will be B. Made of polysaccharide. So again, if we go through those particular options, the only one of those that contains carbohydrate is A, the cell wall, and is an organelle and is not surrounded by two membranes. So knowledge of membrane structure of the different organelles, and remember it's asking for organelles here. So we know that the chloroplast and the mitochondrion have got double membranes, as does the nucleus. So the only one left that contains a membrane is the Golgi apparatus, that is E. So the second question says that human breast milk is produced and secreted by gland cells. These gland cells have adaptations that include many mitochondria and Golgi vesicles. The milk contains high concentration of protein. Explain the role of these cell adaptations in the production and secretion of breast milk. So we've got to link the mitochondria and the Golgi vesicles to the production and secretion of breast milk. So if we start with mitochondria, we know that whenever there's large amount of mitochondria in a cell, then that cell requires large amounts of energy. Mitochondria is associated with energy. So what we can say is that the mitochondria release ATP we've got to explain why the ATP is required. So it's not just good enough in a question like this to say, well, the mitochondria release ATP. We've got to say how those ATP are going to be used. So in this case, it's for the movement of vesicles. And the synthesis of proteins. Some students for this question talked about producing energy. Remember, we don't produce energy. We can't make or destroy energy. We can only convert it from one form to another. So that's why we can say that they release ATP, because ATP is a form of stored energy. So a lot of students did sort of like, do well in this part of the question, talking about mitochondria, but the role of the Golgi apparatus was less well known. So for this, we can say that the Golgi vesicles transport the protein out of the cell. So once the proteins have been manufactured, we need a way of getting them out of the cell, and that's the role of the Golgi vesicles. Question 2 asks us to describe how a peptide bond is formed between two amino acids to form a dipeptide. So the first thing we need to know about what happens during the formation of a dipeptide bond is the type of reaction that takes place. And the type of reaction that takes place to form a peptide bond is a condensation reaction. So this is a type of reaction where we lose a water. That's what it means by condensation. So we can say condensation reaction. You could have got away with a reaction where water is lost. And because it's worth 2 marks, we need a bit more detail. In this case, we need to say what parts of the amino acids are involved in the peptide bond. So the peptide bond forms between the amine and carboxyl groups. Right, part 2 asks the secondary structure of a polypeptide is produced by bonds between amino acids. So here we've just got by bonds. It's not telling us what type of bonds those are. So obviously we've got to say what bonds are necessary to hold the protein in particular aspects of secondary structure. So the type of bonds involved in secondary structure are hydrogen bonds, and again we need to say where the hydrogen bonds are in essence what parts of each amino acid are involved in hydrogen bonds. So the hydrogen bonds are between the NH groups of one amino acid and the C double bond O group of another. So we could also talked about the types of secondary structure that are formed to create that are created by the different types of arrangements of hydrogen bonds. But the key is to be precise. You can't just say the hydrogen bonds exist between one amino acid and another. We've got to actually explain which groups are involved. Right, part 3. Two proteins have the same number and type of amino acids, but different tertiary structures. Explain why. So all it's telling us here that they've got the same number and the same type of amino acids. It's not actually saying that it's the same order. So we know that the order that amino acids are in is the primary structure. So we can say that they could have different primary structures, and because they're going to have different primary structures, then the amino acids that can be involved in hydrogen bonding or ionic bonds are going to be in different places. So that means that the ionic bonds and the hydrogen bonds will be in different places in the protein. So they're going to be further along or more closely together than another protein with the same amino acid content, but those amino acids are in a different order. So we can say that because they have different primary structures, that they form ionic and hydrogen bonds in different places. Now, during the exam, a lot of students got confused about the structure of proteins and DNA. So again, going into the exam, make sure you know the different types of structures and the different types of bonds that are involved either in protein structure and DNA structure. So question 3 asks us to describe the relationship between size and surface area to volume ratio of organisms. This is a relationship that you should have encountered quite a few times during your studies, both at A-level and GCSE. So we know that as size increases, the surface area volume ratio decreases. So the next part involves a calculation. So we're told that a scientist calculated the surface area of these eggs, which are spherical, and the surface area is 9.73 square millimeters. We're given the equation for surface area, and we're given a value for pi. So we're asked to calculate the mean diameter of the frog egg. So first thing we need to do is rearrange this equation. So we've got R as the term. So R becomes root surface area divided by 4 pi. So that becomes root 9.73 over 4 pi. And that comes out to be R equals 0.8801. But remember, in the question, we're asked for the diameter, not the radius. So we just need to double the radius to get the diameter. So the diameter becomes 1.76 meters. So the key there is, again, read the question and make sure you're giving the right parameter that you've been asked to give. In the next part of the question, we're given some data to do with the ratio of surface area to mass and the mean uptake of oxygen in micromole per gram per hour. It says the scientist used the units of micromole per gram per hour for the rate of oxygen uptake. Suggest why you used micromole in these units. Now, obviously, the amounts of oxygen that are going to be taken up are going to be really small amounts because these are small organisms. So, because they're such small amounts, rather than use really small values with lots of decimal points or using standard form, if we use micromole as opposed to mole, then we're going to get whole numbers rather than these fractions. So the reason that he used micromole was due to small amount of oxygen uptake avoids use of standard form. Or you could have put avoids the use of numbers with many decimal places. So within this question, over 60% of students scored 0, either because they talked about the organism being too small or talking about accuracy and precision. And again, using a different unit or a smaller unit like micromole doesn't affect the precision or the accuracy. You're using exactly the same numbers, you're just not using the standard form or decimal places. So the next part of the question says the scientists decided to use ratio of surface area to mass rather than the ratio of surface area to volume. Okay. He made this decision for practical reasons. Suggest one practical advantage of measuring the masses of frog eggs, tadpoles and adults compared with measuring their volumes. So when you've got something that's a very unusual shape or non-standard shape, so it's not a cube or a sphere or things like that, it's very difficult calculating volumes. But calculating mass is relatively easy because you just put them on a balance. So for practical reasons, it's easier because of regular shapes. Therefore, difficult to count volumes. Says next, explain why oxygen uptake is a measure of metabolic rate in organisms. Now here that we know that oxygen is used in respiration, but that doesn't tell us about metabolic rate. We've got to tell us why an increase in oxygen, therefore an increase in respiration, how that is linked to metabolism. So we need to put that oxygen is used in respiration, which provides energy for those metabolic processes. So again, you've got to link the oxygen uptake with the actual metabolic processes which need energy. So again, if students who just put because it's used in respiration, they're not going to get the marks because they've got to link it to the metabolic rates. So finally in this question, it says a student who looked up these results said that they could not make a conclusion about the relationship between stage of development and metabolic rate. Use the information in table 2 to explain the reasons why they're unable to make the conclusion. So first off, if we go back and look at the table, there wasn't any information about the uptake of oxygen for the eggs. So we can't make any conclusions, can't draw any conclusions to do with that stage. You can't compare all of the stages. So that's the first thing we can put, that there was no information about the egg. So we can't compare all stages. So that'd be enough for 2 marks. But the third one, although we've got the mean rates, we don't have any statistical information, so we can't see the standard deviations. So if the standard deviations are really big, then there may be an overlap between the numbers of one stage and a number of the other. So then you can't make these comparisons. So just looking at the value of the mean rate, it looks like the adult is much lower than the tadpole, for example. But without knowledge of the standard deviation, we don't know whether actually, when you take into account the standard deviations, they may be an overlap or really close. So for our third mark, we can say that there is no statistical information. Therefore, no comparisons of standard deviations. So question 4 asks to give two similarities in the movement of substances of diffusion and biosmosis. So again, two processes that you should be familiar with. So we know that in both cases they have a movement from a high concentration to a low. So in the case of diffusion, this might be a high concentration of a solute to a low, and in the case of osmosis, it's the movement of water from a high concentration of water to a low concentration of water. The other thing about both processes is they don't require energy, so it's not an active process. So we're then given a graph to do with the movement of monosaccharides and monoglycerides, where we've got external concentration along our axis and the rate of uptake of whether it be the monosaccharide or the monoglyceride by the epithelial cells. So the first question to do with this graph says, use your knowledge of transport across membranes to explain the shape of the curve in figure 1 for uptake of monosaccharides between concentrations A and B. So we can see that in A and B we've got a slight curve, but generally it's quite a straight line. But that's indicative of a process where that the movement is through carrier proteins, and because generally the rate of movement is going up as we have more a higher external concentration, that's showing that not all of the carrier proteins have reached saturation. So we can say for parts A and B, between A and B, that the movement is through carrier proteins, and the rate of uptake is proportional to the external concentration. Now between C and D, it's leveling off to reach a constant rate. So here we can say that all the channels are saturated. So the final part of this question says figure 1 is evidence for monoglycerides being lipid-soluble molecules. Suggest how. So we can see that in the graph it's just a straight line. So as the external concentration increases, so does the rate. Okay, so that is suggesting diffusion because it just keeps going. The more concentration, then it's directly proportional to the rate of uptake. So we can say that as the rate of uptake is proportional to the concentration, then diffusion is taking place. Now, if it's diffusion, what that means is there's no carrier proteins, and these molecules are going straight through the membrane. And we know that the membrane is a phospholipid membrane, so the inside of the membrane does have lipids. So the fact that these molecules are managing to go through the membrane, it must mean that they are lipid soluble. So therefore, the lipid molecules are diffusing through the phospholipid bilayer. So question 5 tells us how a student prepared a stained squash of some cells from a tip of an onion root and observed it in an optical microscope. So it says during the preparation of the slide he cut the first 5 millimeters of the tip of an onion root and placed it on a glass slide, then covered the tip with a drop of stain solution and a cover slip. Warmed the glass slide, then pressed down firmly on the cover slip. He identified and counted nuclei in different stages of the cell cycle. Explain why the student used only the first 5 millimeters of the tip of an onion root. So if the student wants to identify different stages of the cell cycle, he needs to see the cells that are dividing, and it's only in this region do you get cells dividing. So we can say they only used the first 5 millimeters because this is where dividing cells are formed. Press down firmly on the cover slip. Now a lot of students for this question put about removing air bubbles or distributing the stain. That's not the case. The reason that we press down firmly on the cover slip is to make sure that we've got a thin layer of cells, because if we have lots of cells on top of each other, then there's going to be no light that's able to get through. It's going to be too dense. So we press down on the cover slip so that there is a thin layer of cells so light can pass through. So the next part of this question requires a calculation. So we're told that scientists have found the mean length of time spent by onion cells in anaphase of mitosis to be 105 minutes. They also found the cell cycle of cells in the onion root, shown in figure 2, takes 1080 minutes. So we've got 32 whole cells shown in figure 2, and says use the information in figure 2 to calculate the length of time the cells of this onion root are in anaphase, and then calculate the percentage difference between your answer and the mean length of time found by the scientists, which we've been told is 105. Show you're working. So we know that there's 32 cells in this picture. So we've got to, first of all, work out or identify which cells are in anaphase. So if we look at that, we can see the cells that are involved in anaphase are these 3 here. So out of the 32 cells present, there's going to be 3 in anaphase. So the fraction that are in anaphase is going to be 3 out of 32. And we know that we've been told that the entire cell cycle takes 1080 minutes. So it's going to be this 3/32 fraction of the 1080 minutes that is the time spent in anaphase. So we take all fraction that are in anaphase, multiply it by the full time of the cell cycle, which is 1080, and that comes out to be 101.25 minutes. And we've been asked to find the percentage difference between our answer and the mean length of time. So the percentage difference, first off, we're going to have to find the difference between our time and the scientist's time. So that's going to be 105, which was their time, minus our time, which is 1 or 1.25. And we divide it to be able to get the percentage difference by their time, which is 105. Multiplied by 100, so we get a percentage, and that comes out to be 3.57%. So next part is a multiple choice question, and we're asked to tick the name given to the cell, to the division of cytoplasm during the cell cycle. And again, these are some of the definitions that you need to know, and the division of cytoplasm during a cell cycle is called cytokinesis. We're then asked to describe and explain what the student should have done when counting cells to make sure that the mitotic index he obtained for his root tip was accurate. Now again, like all of these procedures that involve counting, you want as large a sample area as possible to make sure that you've got a representative sample. So for this one, we can say examine a large number of cells so to ensure a representative sample. So the final part of this question says, a scientist treated growing tips of onion roots with a chemical that stops roots growing. After 24 hours, he prepared a stained squash of these root tips. Figure 3 is a drawing showing the chromosomes in a single cell observed in the squash of one of these root tips in anaphase. The cell was typical of other cells in anaphase of these roots. So what's happened is we can see that we've got sister chromatids, but they've not been separated during anaphase. The anaphase has been stopped, and the reason that they have not been separated is because the spindle fibers haven't been formed. So what we can say, it says use the information to suggest how the chemical stops the growth of roots. So we can say that it stops the anaphase by stopping spindle fibers forming, which prevents separation of sister chromatids. Question 6 describes some experiments to investigate substance X on the growth of new roots. So the diagram shows the 4 different experiments and how the substance is applied and how the stem differs in each one. And we've got a table of results showing the mean number of roots per length of stem in each experiment. So part 1 says treatment D is a control. Explain how the measurement obtained from this control is used by the scientist. So if we have a look at what D is, so we've still got a block of agar at the top, but there's no substance X in the agar, and the middle section of the stem is intact. So we need to know why, or explain why the measurement obtained from this control is used by the scientist. Now what the scientist wants to do is know how effective substance X is, and also the things that may affect the effectiveness of substance X. So by using an untreated piece of stem, so a stem that doesn't have any substance X on it, then they can use a direct comparison to say whether substance X has had a positive effect, has had a negative effect, or has had no effect whatsoever on the growth of stems compared to a root that has no substance X applied. So what we can put for this is that they use D as a control to, so it's used to compare the effect of other treatments and it shows the effect of substance X compared to substances produced naturally. Part 2 says using figure 4 and table 3, what can you conclude about treatments D and E about root growth? So if we look at treatments D and E, so D we've already discussed that it's got the agar but no substance X. Treatment E is where we have substance X in the block of agar and we've got a full stem, and there's been no other things applied to the stem. And if we look at the results, we can see that the mean number of roots per length of stem has doubled. But note that even without treatment X, we're still getting a number of roots per length. It isn't that substance X is giving us roots and without substance X we're getting no roots. We're just getting more. So for our answer, we can say that D shows us that substance X is not required for root growth. Because we're still seeing roots grow without any substance X there. We also know that substance X moves through the plant. We can tell this because the agar is right at the top of the stem, so it must be going into the stem and going through the plant to produce those roots in the plant. And E shows us that substance X increases root growth. So as we saw that there were more roots in E than there were D. But also note I've shown that I've mentioned D and E. So the conclusions that we're drawing, I've indicated which diagram I've drawn those conclusions from. Right, 6.3 says the mass flow hypothesis is used to explain the movement of substances through the phloem. Evaluate whether the information from this investigation supports this hypothesis. Do not consider statistical analysis in the answer. Okay, so what we've got to do here, because we're evaluating, we've got to come up with some points that support and are against the actual hypothesis. Okay, so I'm not just saying yes, all the evidence points towards it. We are critically evaluating it, so we've got to give some plus points and some negative points. So we can say that in support, that F shows that the phloem is involved. Because when the phloem's been removed, we can see significant less roots that are grown with substance X applied. And G shows that active transport is involved as cooling reduces movement. So in G, we know that it's the intact stem, and all we've done is cool it. And by cooling it, also we're slowing down chemical reactions. We're slowing down, the release of energy, and that in turn is slowing down the movement of substance X, and we're not seeing a significant amount of roots grown compared to the control. So against the hypothesis, we don't see any bulge above the ringing in F, which we would see, as part of the hypothesis, the mass flow hypothesis. So we don't see above ringing in F, and the roots still grow without phloem. So we can see that when the phloem is removed, we're still getting some root growth. So it's showing that the substances that produce root growth aren't necessarily going just by the phloem. So roots still grow without phloem. Question 7 starts by asking us, what is digestion? Now, obviously this is an AS paper, so we want enough information to a higher level for AS level. We don't want things like it breaks down food, or some answers will talk about it actually being absorbed by the gut. We've got to be specific about what digestion is, and the type of answer that we'd expect at AS level, it's the hydrolysis of large substances to smaller substances. And against one of those definitions that you will find in your specification. So part 2 says one species of fungus digests cellulose using two types of enzymes, endocellulases and exocellulases. Endocellulases act in the middle of the cellulose molecule, and exocellulases act at the ends. Endocellulases and exocellulases act at different parts on cellulase molecules, suggest why. Now these are enzymes, and like any enzyme that shows specificity, which these do, whether it be from the end or the middle, it's all to do with the active sites of the enzymes being complementary to the substrate and having specific shapes that will bind those substrates. So it's exactly the same here. So we can say that the active sites of the molecules are different shapes. So therefore, complementary to different parts of cellulose. So the next part involves a calculation. So it says a scientist prepares the following mixtures. So in each case, we've got 15 grams of cellulose, and we've got the same concentration of whether it be the extracellulose, the endocellulose, or a mixture of both. And then we've got the results of how much cellulose is left after 24 hours. So the question asks us, use the information in the table to calculate the rate of digestion of cellulose when both enzymes are present. Give your answer in grams per minute and in standard form. It says show you're working. So the first thing we need to know is how much cellulose is being digested. So we saw earlier up in the question, we start with 15 grams. So if we start with 15 grams and we end with, in this case, 9.2, when both cellulases are present, and that gives us 5.8 grams that have been digested. But we need to work out the rate per minute. So it's 5.8. Now we've been told that this takes 48 hours. So we need to know how many minutes that contains. So if we put that over the 48 hours times by the 60 minutes that are in each hour, then that gives us 2.01 times by 10 to the minus 3 grams per minute. So the way that we calculate this is how many grams have been digested, and then divided that by the number of minutes that it took to digest it. So the scientists use the same concentration of endocellulase and extracellulase in the mixtures. The rate of digestion of cellulase is greatest when both enzymes are present. Suggests why. Well, in this case, what's happening is we know that the extracellulase can only attack the ends. So if we've just got a piece of cellulose and some extracellulase, then what happens, it can digest from both ends. But if we have endocellulase present as well, then what happens is that one piece of cellulose is cut in the middle, and that produces more ends for the extracellulase to act. So the endocellulase, because it's cutting it up into shorter strands, that produces more sites for the extracellulase to act. So we can put that by saying that the endocellulase creates more ends for the extracellulase to digest. So the last part of this question says the scientist could have expressed a result as a percentage and loss of mass of the cellulose in the space, write an equation for calculating the percentage loss in mass. So we want the percentage loss in mass. So the first thing we need to do is work out what the loss in mass is. So that would be the initial mass minus the final mass. So that will give us the loss in mass. And because we want it as a percentage, we would divide that by the initial mass and multiply by 100. Now, you've got to be precise with the language you use in this. So we've talked about initial mass and final mass. Some students put things like new or old mass or total mass, and that's not really specific enough. It can leave with ambiguity about what you mean by new and old and total. So it's important in questions like this to use very precise language like initial and final. So question 8 is about dilutions, and generally this question wasn't answered well in the exam. So it's important you understand about the dilutions you've carried out during your practicals and be able to place them in other contexts. So it says a student used a dilution series, so I'll just underline that, a dilution series, to investigate the number of cells present in liquid culture bacteria. Describe how he made a 1 in 10 dilution and then use this dilution to make a 1 in 1,000 dilution of the original liquid culture. Okay, so I'll go through how to do this. So the first part to make the 1 in 10 dilution is add 1 part the bacteria culture to 9 parts sterile liquid and mix well. So by adding 1 part to 9 parts, you've got 10 parts in total, and one of those is the bacteria. So it's then how to use this 1 in 10 to get a 1 in 1,000 dilution. So if we've already got a 1 in 10, we need to make a 1 in 100 from this, because the difference between a 1 in 10 and a 1 in 1,000 is a factor of 100. So we could say we take 1 part of this solution that we've made, add it to 9 parts sterile water, and then repeat that. So we've carried further 2 dilutions, or we could say we could add 1 part of this solution and add to 99 parts sterile liquid and mix well to get final 1 in 1,000 dilution. So just run that through again. So because we've taken 1 part and added it to 99 parts, we've got 1 part in 100 of this solution that we made earlier. And because this was already a 1 in 10 dilution, this final dilution that we've made will be a 1 in 1,000. Now only 3.2% of students got all 3 marks for this question during the exam, mainly by not saying mixing, because if you just add 1 part to 9 parts and that's it, you've not really got a 1 in 10 dilution. It's not, the bacteria is not evenly spread, so you've got to make sure that you mix the solutions well after adding them. Okay, part 2 says using an optical microscope, the student determined there were 15 cells in 0.004 cubic millimeters in the 1 in 1,000 dilution culture. So it's the 1 in 1,000, and there's 15 cells in the 0.004 cubic millimeters. calculate the number of cells in 1 cubic centimetre of the undiluted liquid culture. So the first thing to do is work out how many cells there would be in a cubic centimetre of this solution that we're using. So we've got to work out how many 0.004s there are in a cubic centimetre. So the way we do that is all 15 cells times by 1,000, which is because there's 1,000 cubic millimeters in a cubic centimetre, divided by the volume that we're actually using. So in this case it was 0.004. So this is telling us, this part of the equation is telling us how many volume samples that we had it would take to make up a cubic centimetre. Now we know that this dilution of the solution we used was a 1 in 1,000. So to work out how many would be in the undiluted liquid culture, we just multiplied this value by 1,000. And that will give us the amount of cells that would be in 1 cubic centimetre of the undiluted. And that comes out to be 3.75 times by 10 to the 9. Now, one thing that was commented on during the exam, when the examiners were marking it, that students were doing their calculations all over the section of this space that was allocated, and then the answer was hidden within that. So the examiners were trying to search for the right answer. So once you've got the right answer, make sure you underline it or place it in the space provided so the examiner knows what your final answer is. Don't hide it amongst your working out. So now we've got a student looked at cells in the 1 in 10 dilution during his preliminary work. He decided not to use the dilution to determine the number of cells in the undiluted liquid culture. So what he did was he did the 1 in 10, found out that he wasn't able to use this, so then used the 1 in 1,000. Suggest an explanation on the student's decision. So obviously, but if he'd used the 1 in 10 by using the 1 in 1,000, there would have been so many more cells. So in fact, there would have been too many cells in that volume to count. Now, some students said there's too many cells, but you've got to reference why there's too many, and it's all to do with counting. Some students say there's too many cells, therefore time consuming, but again, that is not the issue. It's to do with the count itself. So there's too many cells and the count is unlikely to be accurate. So when you've only got 15 cells to count, then that's an accurate, you're able to accurately count 15. But if it's more likely to be 100 times this, so you've got 1,500 cells to count in that small volume, it isn't likely to be accurate. So again, make sure you are given a reason why, in this case, too many cells would be an issue. So 8.4 says, on some farms, animals are routinely given antibiotics in their food. Scientists investigated whether these farm animals had antibiotic-resistant bacteria in their intestines. They tested the bacteria for resistance to 2 antibiotics, tetracycline and streptomycin. And we've got the results shown in Table 5. Suggest and explain one reason why bacteria resistant to tetracycline are more common than bacteria resistant to streptomycin in these farm animals. So you can see that the number of tetracycline is much higher than the streptomycin. So the reasons for this is, and you've come across this during your lessons, that the antibiotic tetracycline is used much more often. It's the more often used bacteria. So the resistant bacteria are more likely to pass on that allele for resistance to further generation. So again, it's not enough just to say it's because tetracycline is used more often. We've got to explain why there's a high percentage, i.e. there's going to be a high percentage with that resistance allele. So the next part of the question asks us, in recent years, these farm animals have not been given tetracycline in their food, but despite this, the percentage of bacteria resistant to tetracycline has remained constant. Suggest one reason why. Now, obviously, that allele is already there in many bacteria, and that's going to stay there. It's not going to go away. So because it's already there in lots of bacteria, then that's still going to be continued to be passed through to further generations. So we can say that the bacteria pass on the resistance gene when they reproduce. The gene just doesn't disappear because the antibiotic is not used very much any longer. It's still in the population. So the final question on this paper starts with asking us to compare and contrast. So now we've got a compare and contrast question. The DNA in eukaryotic cells and the DNA in prokaryotic cells. Now note it's asking about the DNA, not the cells. Some students during the exam start comparing and contrasting eukaryotic cells and prokaryotic cells. It's not asking for that, it's asking for the DNA in those two types of cells. Now, this question wasn't answered particularly well, and only 0.3% of students got all the marks, and the mean mark was only 0.79. Okay, so very few students could answer these questions, and it showed that DNA structure is not particularly well known among students. So again, make sure you go over this topic before the exam. So because it's a compare and contrast, we can split the answer into two, so we can do some comparisons. So we can say that the nucleotide structure in both DNA and eukaryotic and prokaryotic cells is identical. They have the same nucleotide structure. So we can say Okay, we can also say that the nucleotides are joined the same. They're both joined by phosphodiester bonds. So we can put that as a comparison as well. Okay, so now we can give some contrasts. Now, when it's contrasts, it has to compare one to the other. You can't just mention one of them. You've got to compare it to the other. So I'll give some examples. So for the contrasts, we can say that in general, the eukaryotic DNA is longer than the prokaryotic. So by saying it's longer, we are comparing it to the prokaryotic. We're not just saying it's long, okay, because that's not directly comparing it with the prokaryotic. It could be that they're both long, so we can say that the eukaryotic DNA is longer. Note I'm using longer than the prokaryotic. We can also say that eukaryotic DNA contains introns. Again, just by saying that, it's not enough. We have to compare it with the prokaryotic. So we've got to say that the prokaryotic DNA does not. Now, I know that it seems that the fact that you're mentioning eukaryotic DNA contains introns, you're implying that prokaryotic DNA doesn't, but you have to state it in the answer. Finally, we could say that eukaryotic DNA is linear. But once again, we've got to compare that with prokaryotic. So we can say the prokaryotic DNA is circular. So the important thing when you're contrasting is you've got to use language that is a comparison to the other thing and talk about how the other thing is different to the aspect that you just mentioned. So the final part says that hemoglobins are chemically similar molecules and found in many different species. Differences in the primary structure of hemoglobin molecules can provide evidence of evolutionary relationships between species. Explain how. Now, over 50% of students got 0 marks for this question, and some students concentrated on the structure, the tertiary and quaternary structure of hemoglobin rather than the primary structure that was asked for in the question. And remember, the primary structure is the sequence of amino acids. So first thing is we've got to sort of like link things together. So, how would we get different primary structures, and how could that be linked to an evolutionary relationship? Well, the reason that we would get different primary structures amongst hemoglobins is through mutations in the nucleotide sequence. So we can first of all link what's going to change with how things might change, as it were. So we can say that mutations change the nucleotide sequence, and this causes changes in amino acid sequence. So we've described that how the amino acid sequence changes. It's through mutations in the nucleotide sequence. And how does this happen? So we know that mutations build up over time. So all mutations, if you compared one sequence with another, all those mutations don't happen at the same time. They gradually get more and more. So the more differences in the amino acid sequence, then there's more distance between evolutionary related species. So if two species are very closely related and they've got a common ancestor that's quite close to both, then there hasn't been much time for mutations in the nucleotide sequence to build up. Where if you've got two species that have a very distant common ancestor, so they've diverted from the common ancestor a long time ago, then there's more time for mutations to build up, and therefore their amino acid sequence will be much more different. So to put that down, we can say that more differences in amino acid sequence between distantly related species. So that implies that the distantly related species have an earlier common ancestor. So the earlier the common ancestor, the more time there's been for mutations in the nucleoside sequence to build up, and that in turn causes a larger difference between the primary structures of the molecule.